All right, well, good evening, everyone. Thanks so much for your patience this evening as we have an in-person audience here in Calgary, Alberta, and the traffic just didn't want to cooperate with us this evening, so we were a little bit delayed, so our apologies. For those of you in audience, thank you so much for making the trip down. And I also want to let everyone know that we will be live streaming this research update. So we have an online audience from right across, primarily Alberta and Northwest Territories, but we do have folks right across the country and some international guests online as well. So really want to say welcome to them as well. My name is Holly Bierland. I am the Vice President of External Relations and the Chief Development Officer for Alzheimer Society of Alberta and Northwest Territories, and I'm thrilled to see everyone tonight. We will be talking about sharing details about our research program, Hope for Tomorrow, and really excited to be able to share the outcome of our first competition with everyone. Before I go too much further, I do want to do a land acknowledgement, and I just want to state that myself and the representatives of the Society acknowledge the many nations, First Nations, Métis, and Inuit who have lived in and cared for these lands for generations that we now call Alberta and Northwest Territories. We are grateful for the traditional knowledge keepers and elders who are still with us today and for those who have gone before us. And we make this acknowledgement as an act of reconciliation and gratitude to those whose territories we currently reside on or are visiting. Now, just to get into our program, we have a great lineup for you. Firstly, we're going to hear from our board chair, Alan Tennant, and he'll share some details about the Society with you. And then we'll hear from 3 of our Hope for Tomorrow research awardees. And then we'll hear from George Andrews, who's our Chief Executive Officer for the Society, and he'll share a donor story with us. And then afterwards, we'll have a Q&A session, and the researchers will answer questions. I would ask that you hold your questions until that time, and we'll go back and forth between the in-studio audience and the online audience as well. They'll be able to type a question into the chat, so we'll do our best to answer as many questions as we can. And I'm really excited. We've got some trainees here with us, and trainees are essential to any research program in an institution. And you might see on screen, I'm not sure if it's in view, but they have poster presentations and are available to chat. So our in-studio audience will be able to speak with them live, but for our online audience, what we'll do is we'll do like a quick interview with as many groups as possible, and you'll be able to ask questions of the trainees at that time. So we've got a great lineup. I don't want to delay us any further because we are a little bit behind, but I do want to note that we do have a person who is available to us, a little bit behind here on the slides, and her name is Christine McWilliams. She's on our staff, and she is available for any online support. If you run into an issue, you can contact Christine directly, and her information is on the screen right now, so if you want to take a note of that, if you have an issue, connect with Christine. She's standing by. So without further ado, I want to introduce our board chair, Alan Tennant, and he'll bring some remarks on behalf of the Society. Welcome, Alan.
Thank you, Holly, and welcome everybody. Welcome to Calgary. My wife and I are Calgarians, so sorry to our friends from Edmonton and elsewhere for the traffic. We never know. It's day to day, so today was a bad day. So I appreciate everybody going to the effort to be here. I'm really proud to be the chair of the Alzheimer Society of Alberta and Northwest Territories, and I want to extend a warm welcome on behalf of the Board of Directors and staff of the organization. Thank you so much for joining us here tonight. I also want to acknowledge a couple of our friends from our colleague organization, Barb Ferguson, the CEO, and Chair Lois Wozni of the Alzheimer Society of Calgary are joining us as well. So thank you for joining us. The Alzheimer's Society of Alberta Northwest Territories is committed to making life better for all those living with dementia. We support, educate, and help individuals and families cope with the challenges of this disease. We raise awareness about the stigma associated with dementia and fund research into the causes, prevention, and possible cures for this disease. We advocate for better policies and practices to improve the lives of those living with dementia. We also work hard to build partnerships with health professionals and the community to ensure those living with dementia are receiving the best possible care. We say that we provide help for today through our program, services, information, and resources, while also providing hope for tomorrow. Our hope for tomorrow comes in the form of our support for dementia research. I'm very proud to say that the Society recently made a $5 million investment in Alberta-based research through our Hope for Tomorrow research program. I would like to invite Dr. Marc Poulin to the podium to talk a bit more about the Hope for Tomorrow research program and how it evolved. Dr. Marc Poulin is Vice Chair of our Board of Directors, and he's also Chair of our Research Committee, naturally, and he's worked with our staff and Campus Alberta Neuroscience and other research partners to launch the Hope for Tomorrow research program and this year's grant competition. Dr. Poulin is a researcher himself at the University of Calgary. We're fortunate to have him on our board and involved with the Alzheimer's Society. Please welcome Dr. Poulin.
Thank you, Alan, and welcome everyone, and welcome Barb and Lois. So the Alzheimer's Society of Alberta and Northwest Territories has been raising the alarm for the need for increased research funding for many years. The gap between existing funding and urgent need to research the causes, onset, treatment, possible cures, and quality of life for people living with dementia and their care partners is staggering. Despite the increased incidence of Alzheimer's disease and other dementias diagnoses, research continues to be significantly underfunded. For example, in 2018, dementia received the lowest research funding at $5 per capita. In comparison, heart disease and stroke research received $12 per capita. Multiple sclerosis, $194 per capita, and ALS research, $700 per capita. We have incredible talent across the country and right here in Alberta. Our board of directors really wanted to ensure that we supported research both at the national and provincial levels. Sorry, they told me to bring a thesis here, so. Under the leadership of George Andrews, CEO for the Society, the research committee worked with Campus Alberta Neurosciences, or CAN, commonly known. It announced Hope for Tomorrow research program last year on September 21st, which is World Alzheimer Day. The partnership between the Society and CAN aims to amplify Alberta's capacity for research and community impact regarding the cause, prevention, and cure of age-related neurodegenerative diseases. The Alzheimer's Society of Alberta and Northwest Territories is investing a total investment of up to $5 million over the next 5 years. The funding will be distributed through an annual research competition administered by CAN. Almost exactly a year later, I'm proud to say that we have completed our first research competition and have 5 successful projects to announce. Projects were selected through a rigorous peer review process administered by CAN. And I want to thank CAN for their great support and for helping to make this Hope for Tomorrow research competition a reality. I see René Dumas is here from CAN in the group, and I wanted to acknowledge René and to thank you so much to you and your team for the wonderful work, and we appreciate all that you and your colleagues are doing to support dementia research. You can see on the screen here the 5 recipients of this year's research competition. Congratulations to all award winners and good luck on your projects over the next year. And now on to the guest speakers. Tonight we will hear from 3 of the Hope for Tomorrow researchers And as I mentioned, we will have a Q&A session after the presentation, so please hold your questions until that time. Each presentation will be approximately 15 minutes. So our first speaker is Dr. Bogna Drastovska. She is a postdoctoral associate and a member of Dr. Ganesh's team that received funding for the Hope for Tomorrow program. Dr. Ganesh was not able to be with us this evening. So a bit about Bogna. In 2017, she joined the Assessing Post-Stroke Psychology Longitudinal Evaluation, or APPLE project, at the University of Glasgow in the UK as a PhD student. Her topic was the prognosis of cognitive outcomes following stroke. Working with different data sets, she developed an interest in applying structural equational modeling techniques to allow better accuracy in capturing post-stroke cognitive changes and its complex relationships with various predictors. Having held interim research assistant posts, Bogna joined the Clinical Neurosciences Department. This is an ideal opportunity for Bogna to continue working in research on vascular cognitive impairment with a focus on understanding its course and identifying factors that could be modified to improve cognitive function and preventing decline. Okay, so get ready for the title. It's a long one. The title of the project is TRIC-SVD, a phase 2, double-blind, sham-controlled, randomized clinical trial of a novel research system for remote ischemic conditioning in symptomatic cerebral small vessel disease. Design, methods, and anticipated impact. So please join me in welcoming Bogna to tell us a bit about her talk.
Good evening, everyone. Can you hear me okay? Great. It's a real pleasure to be a part of today's event, especially this is the first presentation I get to do as a new member of the Clinical Neurosciences Department. And I'm incredibly grateful I don't have to read this title out, so thank you so much for that. So I'll just stick to saying TRIC-SVD. So to provide a bit of background information, cerebral small vessel disease is an umbrella term that refers to a number of different harmful changes that affect the small vessels in the brain. And while for a lot of people, at least for some time, these can be asymptomatic, they can actually lead to strokes. As you can see here, about 1 in 5 to 1 in 4 strokes are attributed to cerebral small vessel disease. And this is also one of the leading causes of vascular cognitive impairment and vascular and mixed dementias. So while this disease can have such a significant impact on people's lives, to date there's no approved treatment for cerebral small vessel disease. And remote ischemic conditioning in recent years has emerged as a promising non-pharmacological intervention. And the way this intervention works is you basically have a blood pressure cuff put on either a leg or an arm, as you can see in the picture here, and you inflate it for a few minutes, which restricts blood flow, and then you deflate it, which allows the blood to flow freely again, also for a few minutes, and you repeat these brief ischemic perfusion cycles a few times. And this has been found to have a remote protective effect on other organs, for example, the heart, lungs, kidneys, liver, or for example, the brain, as you can see here. So this effect happens at different time points. So in the first phase, there's a protective effect that starts immediately and lasts for about 2 to 3 hours. And then there's a second phase that develops over 12 to 24 hours and lasts for 2-3 days. But it's also been found that if you apply this treatment chronically, then actually there could be long-term protective effects. So you're probably wondering, how does this actually protect the brain? So that's a good question, and no one exactly knows the answer to this yet. And a lot of what we do know is in studies that focus on other organs, for example, the heart, and also some animal studies. But from what we observed so far, it seems that remote ischemic conditioning activates a few different pathways. One of them is a humoral pathway, which means that different substances are produced and then released into the bloodstream, and that reaches the remote organs and affects them. The second pathway is a neural pathway. So what happens is that nerves that send information from different parts of the body to the brain become activated. And then the third one is a genetic pathway, where the expression of certain genes gets altered, specifically those that are responsible for inflammatory responses. And in turn, it's been found that this kind of treatment can increase blood flow in the brain. It encourages the formation of new blood vessels. It reduces inflammation. And then in case of a stroke, it appears that more tissue survives without permanent damage. And also there's a smaller chance of a recurrent stroke later on. So as you probably gathered from the explanation, so far research in this field is still rather at its early stages. Studies that look specifically at remote ischemic conditioning and cerebral small vessel disease are still very scarce and just involve a small number of people. And there have also been some methodological limitations, which means that it's hard to know how reliable the current evidence is. And there's also been some challenges with the remote ischemic conditioning devices themselves, which I know has actually been something that our group has experienced with a previous trial called TRIC VCI. And what they found is that, first of all, there's very limited technical support, and as we all know, technology does tend to have its glitches. And the other thing was that it wasn't really possible to monitor how the device was being used during the treatment. So if there were any issues, researchers wouldn't find out about it until the device got handed in back after the intervention, and there's pretty much nothing you could do to troubleshoot. So TRIC SVD has been designed in view of trying to overcome some of these past limitations, and the aim of it specifically is to test the feasibility, safety, and tolerability of a new remote ischemic conditioning system. So the primary question for this trial is whether this novel system can achieve at least an 80% adherence rate over 60 days that would justify going into a longer term, going into a longer term, larger scale efficacy trial in this patient population. The other things we'll be looking at is the discontinuation rates of using this device. What is the patient's perspective on using it, particularly if they find it comfortable to use, if it causes any pain, if they find it easy to use. And then also in terms of safety, we'll be monitoring for any serious adverse events, which would include any injuries to arm tissue, any neurological problems, or deep vein thrombosis. And then this is also an opportunity for us to look at, what really is the outcome of interest in these trials. So to see if this treatment can improve cognitive performance, as well as whether there are any changes in blood biomarkers that would be indicative of some of these protective processes that I mentioned a little bit earlier. So our population will be people either 55 years or older, who have evidence of cerebral small vessel disease on brain scans. So you can see an example of a few of these just below. As a psychologist, I've been taught by other people how to look at these, and the good advice I got was to look at anything that appears asymmetrical compared to the other hemisphere, and to look at anything that's too bright or too dark compared to the rest. So for example, in picture A, you can see a lacunar infarct towards the bottom right. And actually, it's also a lacunar infarct in picture C. On the left side, there's like a dark hole, although this is an old one, which is the difference in appearance. And for example, E is an easy one to spot for those who are inexperienced, because there's just these scattered black dots that show microbleeds. So other criteria going back to that is having cognitive complaints or a history of ischemic stroke that's caused by small vessel disease, and of course an ability to provide and willingness to provide written informed consent. Our exclusion criteria are inability to meaningfully converse or no telephone access, so we can still collect feedback from our participants and stay in touch with them throughout the study, and also for safety reasons if they're at high risk of bleeding for any reason. So here you can see an overview of the design of the study. So at the screening visit is when we check for the eligibility criteria, go for the consent process, and we'll also be taking bloods, doing a physical examination, doing the cognitive testing. And here we'll also explain to our participants how the device is used. And at this point, they'll be randomized to either the treatment arm or the control arm. And we're expecting to have 12 participants in each. Then the first follow-up is at 30 days. We'll check for any adverse reactions to the treatment, as well as repeat taking bloods. And also we'll be asking for people to rate their discomfort or pain to do with using this device. And at this point, also people who are in the control arm will start receiving the treatment. So basically, everyone who participates will receive the treatment at some point. And then at 60 days, the second and last follow-up will repeat what happened at the first follow-up, plus some more cognitive testing. And around the end of the study, we'll also be doing some more in-depth interviews and focus group discussions to get a better perspective of how people felt using this device. And between the follow-ups, we're hoping to stay in touch with our participants just over the phone. So in case they're forget any using the treatment or just have any issues, we can troubleshoot as we go along. So this shows you the actual system. On the left, there's a picture of our device, which, as you can see, is actually a bit smaller than a standard blood pressure monitor. On the right is an overview of how the whole system works. Which can be a bit confusing at a glimpse, but the most important things to note here is that the device can be controlled and monitored remotely. So that has a few important implications. So first of all, at the point of randomization, this can happen without the participant or researcher knowing which group someone has been assigned to. Then when people from the from the control group go to the treatment group, this can also be done remotely without having to hand in the device and manually program it. And then finally, we'll be able to monitor device use throughout the whole intervention. So if anything, if there are any problems, we can troubleshoot there and then, and also send people reminders as needed. So the outcomes that we'll be looking at, of course, mimic our key questions. So the most important things at this stage is to look at treatment adherence. So that's the proportion of total planned sessions completed. And for this, we'll be looking at some summary statistics. So for example, on average, what the adherence rate was across all participants, and also comparing the treatment group to the control group. And we'll be similarly analyzing discontinuation rates as well as the pain and discomfort ratings. Then we'll also have our qualitative data from interviews and focus group sessions. So here we'll be looking to look for any common themes emerging for participant responses to see how easy they found to use the device, how comfortable it was for them, and if they have any suggestions of how we could improve it in the future to have better adherence rates for future participants. And then lastly, we'll have our cognitive measures and blood biomarkers. And in case of these, we'll be looking at differences from pre to post-treatment, and once again, differences between the two study arms. So what we hope to achieve for this is, first of all, advance the development of this device made in Calgary, actually, and hopefully it'll overcome some of the challenges that have been found with remote ischemic conditioning devices in the past. And if high adherence is indeed achieved and the device is found to be safe, then this will provide grounds to take this forward into longer-term, larger-scale efficacy-focused trials. And then finally, like with all trials, we hope to learn something from this to improve future studies. Hopefully not just learn from our mistakes, maybe from something that we do well too. And most importantly, from all the feedback we get from participants, and hope that this will help in planning future studies that use devices in a view of improving outcomes for people with stroke and vascular cognitive impairment. So these are just some key references in case anyone would like to look into this a bit further. And that's everything. Thank you so much for listening.
Thank you, Bogna, for a very interesting talk and description of your project. This is working? Yes? Okay. So I'll remind everyone we'll do the Q&A after the three presentations. So we'll move on to our second presenter. Second presenter is Dr. Robert McDonald from the University of Lethbridge. So Dr. McDonald and members of his team have traveled from Lethbridge tonight. It's great to have you here, and thank you for making the trip. I must say that usually the Lethbridge group welcomes me to Lethbridge. We are involved in running a course in Lethbridge, so the physiology department travels there. There's a member traveling once a week, so I've been doing it for about 10 years, and this course has been coordinated by Dr. McDonald for a number of years, and it's always been a great pleasure to be down there to do a lecture with their trainees. So Robert McDonald received his PhD in psychology from McGill University in Montreal. He moved to the Department of Psychology at the University of New Mexico in 1994, completing postdoctoral training on an insert postdoctoral fellowship. He accepted a position at the University of Toronto in the Department of Psychology, where he received tenure in 2000. He returned to Alberta, where he is an associate professor of psychology and neuroscience and a Canada Research Chair at the University of Lethbridge. His current research is focused on understanding interactions among learning and memory systems, the role of memory system dysfunction in the etiology of psychiatric disorder, testing alternative theories of Alzheimer's disease, cofactors contributing to cognitive and motor deficits following stroke, and multiple pathways for memory consolidation. Dr. McDonald's research will investigate 2 gene mutations and a combination of cofactors taught to contribute to sporadic Alzheimer's disease to see how they interact with each other and how the negative effect might be lessened by lifestyle changes. So, Dr. McDonald, I'll turn it over to you for your talk.
Sorry, guys, we actually had Dr. Carr coming up next, but we'll have Dr. McDonald. I just need to slide through these slides here really quick. So avert your eyes so that you don't get any spoilers from Dr. Carr's presentation. And there's Dr. McDonald. Thank you.
Thank you so much for the invitation and for the funding, of course. My talk today is going to be in 3 parts. So briefly, I'm going to describe what Alzheimer's disease is, and some significant discoveries, just a few discoveries that I think are important and have influenced my work. Then I'm going to explain how I got involved in Alzheimer's disease research. It was quite a different approach. And then I'll talk about the research that was recently funded. So Alzheimer's disease is the most common version of age-related cognitive decline. As you know, it's a crippling neurodegenerative disorder, and there's a very strong aging component to it. So aging is a major cofactor. It progresses in stages. So early stages, it's very difficult to distinguish it from other conditions. But in the middle parts, this is the part that most people are most familiar with, is very significant memory impairments that are associated with very specific parts of the brain. And at the end, there's global deficits. There's pathology that's found in the brain of Alzheimer's disease. Senile plaques and neurofibrillary tangles are abnormal accumulations of proteins and protein malformations. There's inflammation that seems to be in reaction to those pathologies. There's neurotransmitter systems that are downregulated. So acetylcholine, for example, a neurotransmitter found in the nervous system, declines every 10 years from about 40 years until you pass away. And neuronal loss, which is probably the main cause of the memory impairments. And if you look at this slide here in green is a plaque, so it's a senile plaque that's stained using immunohistochemistry techniques. And red is a neuroinflammatory marker. And you can see how they congregate around those protein accumulations. So this brain is essentially your brain. The two hemispheres are split apart, and you're now looking on the inner surface of the brain. And in this region here that's highlighted is the medial temporal lobe. And these are the structures of some of the first areas that are degenerated in Alzheimer's disease. And it turns out that these areas are crucial for a very specific type of learning and memory. I got permission to use this photo today. So just to give you an example of what kind of memory impairments Alzheimer's patients have to suffer through. So this, if you showed me this picture and I had Alzheimer's disease deep into the middle stages, I wouldn't be able to tell you that this is my daughter, that she's sitting on a couch in my brother's living room in his place in Sylvan Lake, that it's Christmas in 2009. And also that this little ensemble she's wearing, I purchased in Spain for my son when he was 3 years old. And when I brought it back home, my father-in-law saw the suit and said, "Rob, it's way too small." My son is 3 years old. But luckily we had my daughter a few years later, and it fit her. But this is the kind of thing we think that those structures in the medial temporal lobe, the hippocampus, do. They take all these seemingly unrelated, disparate elements and pull them together into a representation that you can then retrieve later. It's amazing. So Alois Alzheimer is the actual discoverer of Alzheimer's disease. And his first patient was August Dieter. And he did two fascinating things. He did these long interviews with these patients and got an idea of what their functional deficits were. And then when they passed away, he looked at their brain to see if there were any changes. This is a picture of August and some of the original drawings of Alois Alzheimer. And those are tangles that he found in the neurons of her brain. Oddly, in his time, he was not known for discovering Alzheimer's disease. He was known more for his work on syphilis infections and their effect on the brain. Why do you think he wasn't famous for Alzheimer's research? Any ideas? My students might know. Anybody? So we just go back to the date, 1906. Very few people lived to 65. So the disease was seen as extremely rare. Now people are living to 100, 110. And there's tons of Alzheimer's patients now. So this has changed my slides. And my backgrounds and everything are gone. So I guess I'll just wing it. So 3 discoveries I'm going to talk about since Alois. One is this cholinergic theory of Alzheimer's disease. So acetylcholine declining as you age, and some treatments that were developed to test that theory. The discovery of gene mutations associated with Alzheimer's disease, and then finally this massive undertaking to produce preclinical animal models in rodents of Alzheimer's disease based on those genetic discoveries. So Bartas, based on that evidence that acetylcholine was depleted in Alzheimer's patients, claimed that that must be the cause of Alzheimer's. And that led to the development of these drugs that upregulated acetylcholine in patients. You just take a pill, it's going to upregulate acetylcholine. And this is one of the treatments that's currently available. Gene mutations were discovered on 4 different chromosomes, amyloid precursor protein, APOE, and presenilin 1 and 2. If you get any of these, I would avoid presenilin 1. So those discoveries of these genes were amazing, and people said, well, can we induce these in animals and then recapitulate the pathology, the memory impairments? And if we can do that, then we can understand the mechanisms and develop treatments. And this was a heady time. The field was very high on itself. We're going to solve Alzheimer's very quickly. Most of this research, I would argue, is looking at the familial version of Alzheimer's disease. And in this disease, very clear genetic mutations, very clear pathology resulting in memory impairments. Very simple, linear story, leading to the tragic loss of your personal memories. Unfortunately, as people now know, almost 95% of patients get the sporadic version, and the etiological mechanisms are not well understood. So when I was a faculty member at U of T, I had a really influential circadian rhythms researcher on faculty, and I started talking to him, and we wanted to look at the relationship between circadian rhythms and memory. So just a brief explanation. Deep inside your brain, you have a clock, and it's 24 hours. It gets set by the light-dark cycle, and it controls a bunch of different things at different times of day. And what the master clock does is it sets all these peripheral clocks in different parts of your brain and different parts of your body, upregulating things at certain times of day, downregulating them at other times of day. Okay? So what we wanted to do is look at animals, rodents, as they aged and track their circadian rhythms. And what you're seeing on the top here is activity measure. It's called an actogram for these hamsters as they're aging. The dark bars are indicating when they're running on a running wheel, and the light areas are when they're not active. And this is during the day, and this is during the night. They're nocturnal. So we took a huge swath of hamsters and let them age and track their circadian rhythms. And what we found was fascinating. Half of them, their circadian rhythms stayed perfectly intact deep into aging. 80, 90 years old hamsters. Not really, but in terms of human age. And half of them, their circadian system fell apart. So this is really interesting. Then we looked at their memory. We have an assay of episodic memory in these hamsters, and we showed that normal aged hamsters that had normal circadian rhythms had perfect episodic memory. And the ones that were old but had impaired circadian rhythms were terrible. Okay? So this was shocking to us, and it suggested that circadian dysfunction is actually a better predictor of cognitive decline than aging was. So I said, wow, that's interesting. So I went into the literature to see what people were saying about Alzheimer's and circadian, and there was very little. And then I went even broader and looked at all cofactors that have been linked to Alzheimer's disease, and most of those were being ignored as well. So I wrote this theory paper that's not cited very often, I must say. And the basic idea is the following. If you take a large swath of sporadic patients, they will fall into subcategories. And those subcategories are going to be based on the cofactors that are present in those individuals. Different subtypes, different combination of cofactors. So the mechanisms that are driving the pathology and the memory impairments are going to be different, and the treatments have to be different. And we did, for about 10 years, a bunch of experiments where we looked at all these combinations of cofactors. Some of these you'll recognize, some maybe not. So mini strokes, stress, circadian dysfunction, etc. And even some of the protein malformations, type 2 diabetes, etc. And the pattern was quite fascinating. So no effect with some combinations, and that's consistent with the theory. They're not going to synergize. Some combinations produce neurodegeneration that isn't there on their own and produce memory impairments. And some produce memory impairments but no neurodegeneration. And that suggested to us that the mechanisms in those subtypes must be different. So that work and that theory landed like a lead balloon for about 20 years. So no one was interested in it. It wasn't Alzheimer's research. But very recently, there's renewed interest in the sporadic version now. And the reason is these discoveries of these genetic polymorphisms that are found in the sporadic population. So these are genome-wide association studies. And what they found is a bunch of polymorphisms that sort of fall into categories. Excuse me. Inflammation, lipid metabolism, immune responses, and endocytosis. And I suspect there'll be more. So our theory has not changed. The only difference now is these gene polymorphisms are probably susceptibility cofactors. Okay? Just changed again. I wanted to note sex differences as well. So it appears that women are more affected by Alzheimer's disease than men. It's very controversial. But there's been some really interesting work suggesting that females are more susceptible to particular gene polymorphisms. So if they get a particular polymorphism like APOE4, they're more likely to descend into dementia than men. So there's really interesting incident rates differences, disease presentation differences, and symptoms, and that suggests to us that the etiologies are actually quite different in men and women. And it is not well understood at all. So finally, our research. So we're going to try to develop animal models, rodent models, of these SAD subtypes. We're going to use both males and females, and we're just going to look at the impacts of the gene on its own, the gene polymorphism on its own, do a deep dive into the changes in the brain and the body, and then we're going to look at cofactors and what impact they may have. So lifestyle factors, for example. And then if we can produce these subtypes of Alzheimer's disease, we're going to try to treat it with exercise and cognitive training. These are the two mutations we're going to look at in this grant. APOE4 is the most common, and it's interesting to us because of its potential impacts on glucose metabolism and vascular function. And MS4A2, which is involved in calcium regulation and immunity. So these animals are going to be produced by Michael Sassner, so we're collaborating with him from Jackson Laboratories. Some of the cofactors, so an unhealthy diet, as you know, high fat, high sugar, has been associated with Alzheimer's disease. We think the APOE4 mutation is going to be particularly problematic for people that have a bad diet. We're also going to look at infections, which have also been implicated in Alzheimer's disease. And we believe that MS4A2 animal is going to be very susceptible to infections, but not necessarily the diet. Our standard brain-body assessments, cognitive function, we're also going to do metabolomics. So we're going to look at metabolites in their urine, blood, feces, brain, and body tissue, and we're going to relate that to the changes that we see in pathology in these different types of animals. And we're hoping that this is going to give us some novel biomarkers and potential improvements in early diagnosis. This is Dr. Tony Montina, who we're collaborating with. He does lots of metabolomics in Lethbridge. Treatments, exercise, which is the best treatment we have available right now, as Mark knows. And we're also going to look at cognitive training. So recently we showed in a familial model of Alzheimer's disease that it was quite effective in reducing pathology and memory impairments, but only when it was multi-domain training, not single-domain training. And the reason we think that is that multiple domain training is activating different networks, whereas single domain is probably activating just one. So these are the designs: gene polymorphisms, memory, pathology, gene polymorphisms, some lifestyle factor, in this case a diet, memory, pathology, and then same experiments again, but this time with the treatment. Predictions, we think we're going to provide evidence for these subtypes of SAD. Specific treatments will work for some subtypes and not others. Sex differences will add a layer of complexity. And the impacts, better understanding of SAD, it's not well understood. We're working with researchers in Montreal who do similar work in humans, and we're going to compare our results from the human and the rodent, excuse me. And then what we think ultimately, if this kind of work is continued, is there'd be some kind of genetic testing early in middle age that would help you sort of assess people's genetic polymorphisms, and then we can provide particular lifestyle factors to avoid and preventions. This is our team currently. Nyung, Danica, Malia, David, Beirut, Abby, Claudia, Matt, Amanda, Baron, Tina, Dean, Vanessa, Haley. Thank God for my hippocampus. Thanks to the organizers tonight. Really appreciate your work. CAN and Alzheimer's Society of Alberta and Northwest Territories, of course, and most importantly, the donors.
So I guess we'll have to go backwards now. So thank you very much, Rob. I'm excited to see what you will find. It sounds very interesting, and there's a saying that goes that everything that gets worse with aging gets better with exercise. And in fact, there's a few studies, notable studies that were published in journals like The Lancet recently that have shown that there's a number of lifestyle factors across the age span that play a role, and to this day exercise seems to be the only disease-modifying intervention for Alzheimer's disease and other dementias. So very interesting talk, and good luck to your team. So I'd like to move on to our third speaker, Dr. Satyabrata Kar from the University of Alberta. Dr. Kar joined us from the University of Alberta this evening, and we really appreciate you traveling here to be with us tonight. So Dr. Carr is a professor in the Department of Medicine and Neurology at the University of Alberta. After completing his undergraduate studies and first PhD on snakes and frogs in India, he did a second PhD on the spinal cord in 1990 as a Commonwealth Scholar at the Hammersmith Hospital at the University of London in the UK. Subsequently, Dr. Carr did his postdoctoral training on growth factors with Dr. Rémi Quirion at McGill University in Montreal. In 1998, Dr. Carr joined McGill as an assistant professor in the psychiatry department. In 2003, he moved to the University of Alberta. Over the last 2 decades, he has been working to understand why selected groups of neurons are affected in Alzheimer's disease pathology and how to protect them. His current research focuses primarily on biodegradable nanoparticles as novel therapeutic reagents for the treatment of Alzheimer's disease. Dr. Carr has more than 100 peer-reviewed publications and has received many awards and recognitions, including the Young Investigator Award from the Canadian College of Neuropsychopharmacology, Chercheur Boursier Award from Fonds de la Recherche en Santé du Québec, and a Tier 2 Canada Research Chair and Senior Scholar Award from the Alberta Heritage Foundation for Medical Research. More recently, he's been inducted as a Fellow of the Canadian Academy of Health Sciences. He has created and taught multiple courses for undergraduate and graduate students, and many of his own trainees have gone on to successful careers in academia and biotechnology. His research program over the years has been supported and by several organizations, including the Canadian Institutes for Health Research, NSERC, Alberta Heritage Foundation for Medical Research, University Hospital Foundation, and the Alzheimer's Society of Alberta in the Northwest Territories. Dr. Carr's team will be studying PLGA nanoparticles as a potential way to prevent memory impairment by stopping the interactions of proteins believed to cause brain cell death for possible future treatment for Alzheimer's patients. So I'd like to invite Dr. Carr to come and tell us about his work.
Hello. Thanks, Dr. Pule, for the introductions. And thanks also including me as a speaker for these symposiums. And the titles of my presentations, as you can see, is a novel nanoparticle-based treatment strategies for Alzheimer's disease. And all the data, what I will be showing it to you, it has been generated in the last 3 years. And it is a completely new field. I've never worked on it before, so there are a lot of gaps you may find it. Okay. So for the sake of convenience, I think Dr. Sutherland has already mentioned, and Dr. McLeod has also mentioned Alzheimer's disease. What I can do it, a brief introductory note about Alzheimer's disease and how accumulations of a protein called beta amyloid peptides can induce cell toxicity and subsequent development of disease pathology. After that, I'll show you some of our recent data, how a molecule called PLGA nanoparticles, which have been used in delivery of drugs in humans since last 20 or 30 years, can have effects on A-beta aggregations and toxicity, and can also reduce pathology as well as cognitive deficits in animal models of Alzheimer's disease, and also can attenuate cellular pathology in neurons derived from AD patients. And also, more recent data, I'll show you how these nanoparticles, when it is labeled, can be used as a diagnosis for the pathology. So Alzheimer's disease, as you all know, is a progressive neurodegenerative disorders characterized by gradual loss of memory followed by deteriorations of higher cognitive functions such as language, praxis, judgments, calculations. The disease afflicts approximately 6% of the populations over 65 years of age. That is not important. Importance, Dr. McDonald told me that it doubles every 5 years thereafter. if you calculate it, approximately 35 to 40% of the people living over 85 years of age may suffer from this devastating disease. Etiologically, if you look at it, like many other neurodegenerative diseases, Alzheimer's disease is also heterogeneous. Majority of the cases are sporadic, whereas only minority of cases, which is less than 10%, they are caused due to the mutations of 3 known genes. One is an amyloid precursor protein, another is a presenilin 1, another is in presenilin 2 genes. The features or the characteristic features that are associated with Alzheimer's disease includes gross brain atrophy. This is your normal brain, and this is a brain from Alzheimer's patients. As you can see, the atrophy of the brain overall. And loss of neurons in selected regions, not everywhere, only in selected regions. And the presence of neuritic plaques, these brown color structures in the brains, which contains the beta amyloid peptides, and also the presence of neurofibrillary tangles, these violet arrows, which contains another protein called tau. Unfortunately, at present, there is no cure for Alzheimer's patients. And the drugs that have been approved, such as donepezils, rivastigmine, galantamines, or memantine, they provide symptomatic relief only for a subset of AD patients. And the efficacy of the newly approved beta monoclonal antibodies remains to be determined. Several lines of experimental evidence gathered over the last 2 decades indicate that prolonged increase in the levels of these beta amyloid peptides can trigger its aggregations, which can subsequently cause the death of neurons and then development of dementia and disease pathology. Given the critical roles of these molecules, majority of the drug development strategies are targeted, majority, not all, majority is targeted to decreasing the beta productions and its aggregations, or inhibiting the toxicity induced by these peptides, or increasing its clearance so that you can reduce the level of the peptide. However, one of the major limiting factors in developing effective treatment strategy for Alzheimer's disease is the presence of a barrier called blood-brain barrier, which inhibits the entry of any effective drugs or agents into the brains. Over the last decades, what happens? Various nanoparticles, which are engineered materials measured about less than 100 nanometer in length, have been used to overcome these protective blood-brain barriers to deliver the drugs into the brains, which can influence beta aggregations, toxicity, etc. In fact, these PLJ nanoparticles, which are currently being used in delivering drugs in a varieties of conditions in human, when those nanoparticles are conjugated with certain drugs like donepezils or curcumin, they seem to have a beneficial effects in cellular as well as in animal models of Alzheimer's disease by inhibiting beta toxicity and aggregations. It has been shown. However, nothing is known how native PLJ nanoparticles, that means without conjugations with any drug and agent, can influence Alzheimer's disease pathology. That's what we did. We evaluated the effects of these native nanoparticles on beta aggregations and toxicity in silico, that means in test tubes and also in neurons grown in culture conditions, and its effects in animal models of Alzheimer's disease, and also its effects on neurons derived from Alzheimer's patients. We all know that the formations of beta oligomers, which are small aggregates, and fibrils, which are aggregates of repetitive cross-beta structures, they play critical roles in Alzheimer's disease pathology. There is no doubt. Thioflavulin T is a amyloid dye, which is commonly used in detecting beta amyloid aggregations. When the peptide aggregates, you can able to detect using this dye, because what it does, it produces strong fluorescence emissions upon binding to the cross-beta structures of the aggregated peptides. First, what we did, we first measured the aggregations properties of beta amyloid peptides at the concentrations 1 to 20 micromolar concentrations using TST kinetic assay. As you can see, as expected, these molecules aggregates increasing with increasing concentrations until 4 hours, then reaches the plateau. And these aggregations of the beta amyloid peptides, which have been shown by many others before, the propensity of the aggregations is validated by fluorescence microscopy as well as on the electron microscopic studies. As you can see, the molecules at the 0 hours, they are protofibrils, small, but if you incubate them at room temperatures at 37 degrees centigrade, it starts forming fibril, long twisted fibrils. The conversions of A beta monomers into oligomers and to the fibrillar forms evaluated by another techniques called dynamic light scatterings shows that the molecular structures or the aggregates which are less than 10 to 100 nanometer at the early stages, it becomes bigger and ranges to 10 to 10,000 nanometer in size, which indicates that the presence of high molecular weight molecules following aggregations. This is also validated by another technique called native page. When you aggregate, when the presence is, this shows the presence of high molecular weight beta amyloid peptides. So this is just the beta amyloid peptide. Then what we did, we performed aggregations kinetic assays of 10 micromolar concentrations of A beta peptide in the presence and absence of different concentrations of these nanoparticles without any drugs or agents. As you can see, this nanoparticle dose dependently inhibited aggregations, spontaneous aggregations of the beta amyloid peptide. And this is validated using fluorescence microscopy, using electron microscopic studies where you can see the nanoparticles are present in close associations with A beta fibers. And this is also evident in your DLS dynamic light scatterings, in your native page analysis where you can see when you incubate, the amounts of the aggregated peptide goes down in presence of the nanoparticle. We also used another techniques, your filter trap assays, where we can able to detect the less amount of aggregated peptides using an antibody, fiber-specific A beta antibody. So this shows that it can inhibit spontaneous aggregation, but that's not important. Important is that in Alzheimer brains, you do see the molecules are already aggregated. So can this molecule able to trigger disassembly of aggregated molecule? For that, what we did, we incubated preformed A beta aggregates with 2.5 to 50 micromolar concentrations of this nanoparticle. And when you do that, over 120 hours, it triggers disassembly of aggregated aggregated beta amyloid peptides. This is evident in the TST kinetic assay, also in the fluorescence microscopy, electron microscopic studies, also in the DLS and in the native pages. As you can see, the aggregated fibers are breaking down, and you'll see the reduced amount of presence of the aggregated in presence of the nanoparticles. So it inhibits the spontaneous aggregations and trigger disassembly of aggregated molecules. Now coming to what is the protective effect? Can it protect the neurons against beta amyloid mediated toxicity? For that, what we showed first, when we expose these primary mouse cortical culture neurons grown in petri dish to the micromolar concentrations of A beta peptides for different period of times, you can induce toxicity. The cells, they do die. And that is evident by reductions in a marker called MTT, it goes down, increase another marker called LDH, it goes up, following incubations with beta amyloid peptide. I'm not sure whether you can see, also evident, significant increase in the presence of red labeled dead neurons. So that means when you expose these cells to the beta amyloid peptide, they do die. And that's how is the mechanisms? We saw that, we also observed that you will see an increased levels of the carbonylated protein, which is a marker of the oxidative stress, increased activations of some of the kinases, tau kinases, phospho-ARC, phospho-GSK, increased phosphorylations of the tau protein, increased cleaved caspase 3. That's the usual mechanisms many people have shown before. But interestingly, what we found, when you treat these cells with beta amyloid along with these nanoparticles, both pre-treatment and co-treatment, they can able to protect them against toxicity. They are not going to protect them 100%, but significant amount. And that's mediated by reducing the signaling mechanisms, reducing the levels of the carbonylated proteins, tau kinases, tau phosphorylations. As a follow-up to the cultured neurons, we wanted to determine, can these PLJ nanoparticles able to reduce AD-related cognitive deficits and A-beta pathology in an animal models of Alzheimer's disease? For that, what we did, we chronically administered these nanoparticles into the brain of an animal model called 5XFAD mice for 28 days using Alsat mini-osmotic pump. After that, we measured their cognitive performance using these two tests, novel object recognitions and Y-maze test. As expected, these are the control mice, control wild-type mice, wild-type mice treated with the PLGA is the control mice, control 5XFAD mice which are treated with the CSF, no drug, just CSF as a control, and this group of mice treated with the PLGA. As expected, the control 5XFAD mice, that means which are treated with the CSF, they do exhibit cognitive behavioral deficits in both the test, novel object recognitions and spatial memory. But PLGA treated 5XFAD mice, you can able to reverse the cognitive deficit in both the test, in object recognition as well as in Y-maze test. Subsequently, we showed that the levels of the beta amyloid peptides which are markedly increased in these animal models of Alzheimer's disease, it is significantly reduced in affected regions in animals treated with the nanoparticles. In parallel, the number, size, and areas occupied by the beta amyloid containing neuritic plaques, which are the characteristic features of the Alzheimer's disease, are found to be significantly reduced in PLG treated 5XFAD mice compared to the untreated mice. This is the control brain sections where you don't see presence of beta amyloid containing neuritic plaques as expected. So what it shows that the PLG treatments can able to reverse the cognitive deficit partially and also able to attenuate A-beta pathology in animal models. Finally, we used neurons, iPSC neurons derived from the AD patients and control patients, control individuals to determine if PLGA or to investigate if PLGA can able to attenuate AD-related cellular pathology in these cells, and can this cell be protected against beta amyloid mediated toxicity when you use these nanoparticles. These are the neurons derived from the controls as well as in the AD patients. And interestingly, when we treated them with the PLG nanoparticles, the increased levels of the A-beta and the APP seen in these neurons derived from the AD patients found to markedly decrease. So it decreases the levels of A-beta and also precursors protein APP in neurons derived from the AD patients. The neurons from AD patients and control individuals, if you expose them to the beta amyloid peptide, they also will die like mouse cultured neurons. Interestingly, when you treat them with the nanoparticles, they are found to be significantly protected against beta amyloid mediated toxicity. And there is a signaling mechanism is there. It is protected by attenuating your tau kinases, phosphorylations of the tau proteins, and cleavage of the caspase 3. So that means it shows that these nanoparticles can able to protect the neurons derived from the AD patients against beta amyloid mediated toxicity. Finally, so given or considering the evidence, these nanoparticles can able to suppress A-beta aggregations It is of clinical interest to determine if labeled PLJ nanoparticles can able to detect beta amyloid containing neuritic plaques in 5XFAD mice. Because it is binding, that's how it's inhibiting its aggregations. So can this molecule be used in the diagnostic tool? What we did, we injected acutely, in this case just one time for certain period of time, for some period of time, very small amount, labeled PLGA, and then sacrifice the animal 1, 3, 12, 24, 72, 160 hours, then stain their brain sections with beta amyloid antibody as well as congorate, the dye usually used to detect the plaques. What we found, if you look at it, this is only partial pictures. PLJ labeled structures are found to be significantly overlapped with A-beta aggregates which are labeled with the congorate. And this is the PLGA, this is congorate, this yellow color shows the overlap. So, and this is evident at 1 hour, it increases 3 hours, then starts declining after 24 hours. As you can, 1 hour, 3 hour, 24 hours, but after 1 week, you don't see the presence of the PLJ labelings in the brains of these mice, but plaques as expected, it is there. In the wild type mice, you don't see the presence of these particles. So what it suggests that, this is for the first time somebody showing this labeled PLJ nanoparticles can able to recognize the plaques which can be used as a diagnostic tool. So the results obtained so far shows that native PLJ nanoparticle without conjugations with the drugs and agents can inhibit spontaneous A-beta aggregations, can able to trigger disassembly of already aggregated A-beta peptides. It also can increase viability of neurons against beta amyloid mediated toxicity, and chronic administrations can reverse cognitive deficits and attenuate A-beta pathology. And this nanoparticle can protect neurons derived from AD patients against beta amyloid mediated toxicity. And lastly, this nanoparticle when labeled can able to recognize beta amyloid containing plaques at least in animal model. So these results obtained so far indicate that native PLJ may have an unique theragonistic potential in AD pathology. Theragonistic means therapeutic as well as in diagnostic. That's a new terminology. So obviously, I did not do all these works, and I have taken, I have given a snapshots. Many people, I don't have a big lab, I have three or four people. Karthi did most of the in vivo studies, and another postdocs, Bibin and Chi did that in vitro and aggregation biophysical studies. And these are the new people. And I have taken the help of quite a few people. Dr. Fabrizio Giuliani did this, the iPSC cells. Dr. Hogar really helped us in electron microscopic studies. That a lot of helps are there. So, and by the way, thank you for supporting our work. And the works for which I was supported, it is not completely different, it's an extension of this work. Here I am showing the data of beta amyloid. There is another molecule, tau. The project which have been funded, we will look at it, whether it has an effect on the tau pathology or not. Maybe, if we will be successful, maybe in 2 years later, we can talk about it.
Thank you so much, Dr. Carr. I had the pleasure of visiting Dr. Carr's lab, and he was, Marie, I'm just wondering if you could put it to the other slide after the presentations then, so you'll know where they're located. So I had the pleasure of actually visiting University of Lethbridge. We spent some time at Hotchkiss as well, and the U of A, and it was just incredible. There weren't words to describe the depth and diversity of research going on, so it's really quite impressive. So it's so wonderful that we can support some of that great work here over the next few years with the Hope for Tomorrow research program. And now it's my pleasure to introduce Dr. George Andrews, who is going to talk a little bit about the donor impact and share a story. After Dr. Andrews is finished speaking, then we'll go to the Q&A and we'll ask the speakers to come up and we'll get some questions done. Thank you.
Just to be clear, my doctorate's in business, so I was at the edges of a lot of this. But it reinforces to me why this is so important. We talk a little bit about the $5 million, but actually, all of these researchers have agreed to at least match that money. So that impact is $10 million. It's spent in Alberta, and that's purposeful. Now, we can see from this evening's presentation that our researchers and our universities are on par. They don't have to apologize to anybody anywhere in the world with the quality of the research that we're doing here. It's important that we manage that and manage that expectation. And I think we're all kind of singing from the same song sheet, but for me, it's like 1 person in 3 in this province is impacted directly or indirectly impacted by Alzheimer's disease or other dementias. We know that if nothing really happens in the next little while, the next 20 years, we can expect the number of people living with dementia diagnosis to triple, 300% increase over the next 20 years. So the impact on our societies and our healthcare systems goes without saying. So that's why this research is so important. This research is 100% funded by donors. About 90% of our operation is actually funded by donors. So I'm very grateful for our donors, and I don't want to lose any of that. But one of the other things that I wanted to touch base on before I talk about these two gentlemen is that Dr. McDonald mentioned that there is a bias towards female impact with dementia and Alzheimer's disease. But we know from our own study that's about 60%, give or take, a little higher than about 60%. We also know that 2/3 of the informal care partners are female. So imagine that your golden years aren't so golden anymore, or if you're looking after your parents or sometimes spouse and parent, it's a challenge. And so we also know that in 2019, that represented about 40 million hours in Alberta that we were able to document. That's the same as 23,000 healthcare aides working full-time to support people living with dementia. So we need to find a way to wrestle through that process. So in having conversations with these two gentlemen, Buddy Victor and Alex Olston, for some of you in Calgary, you'll know them as local business leaders, they're philanthropists, they're visionaries. In our conversations with them, they got it. They understood the importance of investing in Alberta, living in Alberta, trying to make it a better place for the people living with dementia. They've had a personal journey that ties in closely to this disease, and we're very, very fortunate to have that level of support. So if you are a donor online or in the audience and you're interested in getting involved, we'd love to hear from that, because we've made this $5 million commitment, which will become $10 million, which will become more. The work needs to be done. The work we're doing is fantastic. So on behalf of the society and on behalf of all of the people you will benefit directly and indirectly, I want to thank the researchers and the trainees and the donors for having an impact. And most importantly, we want to acknowledge the great support from Buddy and Alex around how to make this happen. They're visionaries. This wouldn't have happened without their seed money. Thank you.
All right, now we get to an exciting part of the program, and that's the Q&A session. Could I ask the speakers maybe if it's comfortable for them just to come to the front, and then we'll take questions from the in-person audience and online. If you have a question here in person, please put up your hand and we'll run a mic out to you so that everyone can hear. And then we've got someone monitoring online who can help articulate your question here to the crowd. And then we'll just ask folks to answer as appropriate, I guess. Does anybody here in the audience have a question that they'd like to ask, or maybe you need some time to think about it? Yeah, over here? I'll just get the mic over here for you, if that's okay. Please don't be shy.
My question is for Dr. Bogna. I was just wondering what a sham treatment would look like in the case of your RIC treatments.
we'll just try to mimic the actual treatment as closely as possible, which is why this reprogramming is quite useful, because it'll be the exact same device, exact same cuff, but we'll just inflate it to 30 instead of 200 in terms of the blood pressure. So we'll still get the sound probably, and hopefully a little bit of pressure, so it should be hard to tell if you're in the sham or real treatment, but we'll also be asking about that to see if people do seem to be guessing that better than, than we want.
Awesome, thank you.
Thank you.
Maria, are there any online questions at all?
All right, so this particular question isn't really positioned towards anyone in particular, so I'm going to leave it up to all of you guys, and you guys can fight for the mic. Where do you envision Alzheimer's treatment to be in the next 5 years, and then in the next 30 years? Do you think it's going to change, improve? Is there going to be any, breakthroughs in the same way that cancer has seen in the last 50 years? And you guys can do rock, paper, scissors to see who comes up.
I guess the cancer analogy is excellent. So in the US, there was this, they announced, I don't remember what decade, it was going to be the decade of cancer research, and they were going to solve cancer in 10 years. Then the US had that did not happen, and then a decade of the brain in which we were going to solve all of these maladies associated with the brain. And the lesson from cancer is that there's all of these different kinds of cancers, and there's multiple causes, and it's going to take a huge effort to understand each one of those. And I think that's where we're going with Alzheimer's disease. So there's going to be different treatments for different types of Alzheimer's disease as we progress, and a lot of it's going to be lifestyle.
Any questions from in studio at all? No? Great. Oh, good. Just over here.
Hi, I have a question for Dr. Carr. So as you mentioned, these nanoparticles can pass through the brain-blood barrier. So have you tried to inject these particles into the vein and see whether the effect of these particles, instead of just directly inject them into the brain?
That is an interesting question, and that's what we are always facing the problems. The question is that, yes, these nanoparticles, when they are conjugated with the drugs, like any drugs, it does cross the blood-brain barrier. But interesting issue is that very few people, there is some evidence, these native nanoparticles, nobody uses the native nanoparticles. Whether it crosses the blood-brain barrier or not, we don't know. We are studying currently. We do have a very, very preliminary evidence, but in vitro as well as from the in vivo animal studies, it may be crossing the blood-brain barriers. But I cannot be able to say with certainty that it does cross it.
Marie, do you have additionals?
All right, I've got one more here. And again, this is for all researchers. If you were worried that you were potentially going to have dementia, or you're worried about a family member that might develop it someday, whether you're younger or older, what would you guys recommend, based on your research and your general knowledge in the area, that you should do to preempt any kind of early onset?
I think from my talk, I would argue that there's lots of lifestyle factors. So there's lots of things we can control. There's things we can't control. We can't control the genetic predispositions we may have, but we can certainly reduce or improve our glucose metabolism, diet, exercise, stress levels are certainly associated. Type 2 diabetes, so lots of comorbidities are also associated with it. So lots of lifestyle stuff I would recommend.
Yes, I agree. The lifestyle change is one thing. Another thing is that there are quite a bit of studies that are coming out. For examples, one lifestyle change is, of course, we don't use it here in sauna. There is a prospective studies have shown that people who goes for regular to Shauna, they have a less chances of developing dementia. And it is a huge, I think more than 2,000 populations in Scandinavian countries. Apart from that, another, because we are aware of some of the factors, smokings and all those things, it has been affecting our society, and probably some of you might have seen the literatures. Yes, obviously the number of people suffering from the Alzheimer or any related dementia is increasing in absolute number. But the incidence, in certain countries, has started going down. Not everywhere. Certain countries it started going because of the lifestyle changes, regular exercise, and having a good food in terms of the healthy food. Not good food means healthy food like Mediterranean diets and things like that, and keeping your cholesterol levels and other factors in control can significantly affect. There is a issue is that if we can able to delay the onset of the disease by 5 years, we can significantly reduce the number of patients suffering, individuals suffering from the disease. Just if we can delay it by 5 years. And for the first time, I think 2021, until that, we are using the symptomatic treatment. Only in 2021, the first disease-modifying treatment strategies have come out. Of course, there are side effects. And this year also, the second disease-modifying treatment strategies, that is the beta-amyloid antibody, have been used or approved by the FDA. There are side effects, there is no doubt. And that's how the research goes. But at least now we have a domain where we can able to treat the disease, where you can able to reduce the pathology as we progress.
And I'll just do one last call for questions here from the floor, which looks like we can conclude. Oh, go ahead. Brian, who's our producer, has a question.
Yeah, sorry if this has been covered, and it might be obvious, but I don't know. But is there anything that genomics and DNA whole sequence testing can do to indicate like a precursor to you or somebody having Alzheimer's or dementia in the future if you had that data earlier on in your life?
Who's best qualified to answer that question? I don't think I should answer that question.
No, certainly, yeah. So that stuff is being developed right now. So, genome-wide association studies suggest that you have these predisposition genes potentially, and so you should in the future be able to go and get tested, and they should be able to pull out some of these factors if you're susceptible for sporadic version of Alzheimer's disease. Clearly, if you have these familial genes, particularly presenile 1, you're almost guaranteed to get it.
Yes, genome-wide association studies shows that many genes can increase the risk of developing Alzheimer's disease. There is no dementia. But one thing also I should mention here, I believe some of you might have heard there are older adults, some of the older adults who are 80 years old, and data came out in the last 10 years. They do have pathology similar to the Alzheimer's disease, but they don't develop cognitive deficits. And those are, there is a subset of populations, they have pathology almost similar to the Alzheimer's patients, but due to unknown reasons we don't know yet, they don't develop the cognitive deficits. Those are called as an older adults. There is a terminology, I'm forgetting the name. So, and studying of those populations can also enlighten us in terms of the genes that may be involved in triggering the disease. And those genes may not be altered on those populations, older adults, that may lead to the protections against the dementia, development of the dementia. So, and this is just came out, when it came out means, the first paper came out in 2012, I believe, from Northwestern universities. So there is a group of people they live 80, 85, they have pathology, but they are resistant to the dementia, developing dementia.
Great, thank you so much. And that concludes our formal part of the program. And
One more online question.
one more online question. Let's go to that.
Sorry, guys, I wanted to make sure we got all the questions out here. I have, online question referencing Dr. Chen from the U of C Cummings School of Medicine. My phone's being weird here. Made a potential exciting breakthrough with the potential of reversing Alzheimer's, but there doesn't seem to be any support from any pharmaceutical companies. Do any of the presenters know where this research sits? This breakthrough was announced in September of 2020. Do you think it will ever come to a clinical trial? Dr. Chen, Cummings School of Medicine, September 2020.
I think this is our study.
Oh, wonderful. This is why students are fantastic. They know so much.
Yeah, this study is performed in our lab, and we found that the clinical use of the drug carvedilol, which partial of this drug can be benefit for treating Alzheimer's patients. And currently we are trying to push it to clinical trials, and we have got some support from the Greenbow Foundation and also other foundations. But we are still seeking chances to get funded by other people or donors, and we want to put it to the preclinical trials. So we are still working on that. From our studies, are really promising drugs, but we still need to prove it's safe for treating humans. And we do receive lots of email from patients and family members from patients. So they are eagerly to give it a try, but unfortunately, we have to prove it's safe first. So thanks. I think this is one of the patient's families. So we're still working on that one. Hopefully, we will try to make it as soon as possible.
Well, thanks so much for answering the question. That's really great. And again, Marie was mentioning that this is the great part of having trainees in the room and additional experts. So thank you so much for attending tonight. And as I said, this concludes the formal part of our program. But for folks who are here in Calgary, if you want to, we encourage you to mix and mingle, have something to drink, and learn a little bit more about the trainee projects. Some of the trainees are working actively on the Hope for Tomorrow research projects, and others are additional, but there's a real richness in the room, so I encourage you to chat with them and engage and find out more about what they're doing. For the online audience, what we'll do is we'll just move over to the presentations, and each of the trainees will do a short presentation that'll be facilitated, or a quick interview style, I should say, with our communications person, Carrie Candy. So thank you so much for attending formally, and we'll move over to the presentations now, if I can call on Carrie to help with that. Yeah, thanks.
Hi everyone, here's our first trainee.
Oh, I didn't know it was so formal.
Not formal. Can we get your name and what school you're from and the title of your project?
For sure. Hi everyone. My name is Danica Robinson. I'm currently a master's student in the McDonald Lab under Dr. McDonald, and my project centers in around trying to answer the question of, can cannabis extracts treat Alzheimer's disease?
Whoa, cannabis, okay.
Yeah.
We all know what that is.
Right.
Tell us more. What have you discovered?
So cannabinoids are interesting in that they target certain receptors that are in memory centers in the brain, as well as receptors in the immune system. And in Alzheimer's, as Dr. Carr and Dr. McDonald talked about, we begin to see these amyloid beta plaques. And as these amyloid beta plaques kind of grow in size and they increase in number, they trip the inflammatory response or the immune system. And this sort of chronic inflammation essentially wreaks havoc on the brain. It's probably worse or about the same as having no inflammatory response at all. So yeah, I'm trying to decrease the amount of amyloid beta plaques and decrease this inflammatory response that we see in the brain using cannabinoids.
Wow. Well, I can't wait to hear what happens.
Yeah.
Good work.
Well, I do have some results. So I'm doing tasks based on looking at memory in these animals. And some interesting sex differences actually emerge. We're not seeing an effective treatment, but we are, oh, can I? Thank you. But we are seeing some sex differences. And one of my theories as to why that is due to estrogen in these mice. So female mice, they go through what's called an estrous cycle. It's similar to that of a menstrual cycle in humans. And at 9 months, they spend about 3 of their 14 days in this period called proestrus. This is when estrogen is highest. But then at 12 months, they kind of half this, and they're only spending about a day and a half in a 14-day period in proestrus. So my theory is here that they're losing this protective effect of estrogen, and that's why these sex differences are emerging. And then I also looked at their actual brain tissue, and these are the amyloid beta plaques themselves. As you can see here, the positives, that's this group here. There is a trend that the extract is having an effect in decreasing the amyloid plaque number, but unfortunately at this time it's not significant data, but the trend is kind of hinting that we might be going in the right direction. Yeah, thank you.
Wow, very good. Thank you.
Thank you.
Can we talk to the next trainee, please? Hello.
Hi.
Can you please introduce yourself, what school you're from, and tell us about your project?
Yeah, absolutely. So my name is Maliha Panjwani. I am also a master's student under Dr. McDonald in his lab at the University of Lethbridge. This is a particular project I worked on during my undergraduate degree. And essentially, we are investigating the effects of cannabis on the neuroinflammation that occurs in aging mice and other symptoms as well, such as memory, like we've all heard today. So what we kind of did was we took a group of mice that were aged, and we gave them, half of them a mixture of CBD and half of them control mixture. And then we gave them this treatment for about 5 months. And what we did after the 5 months is we ran them on different behavioral tasks that tested different abilities such as memory, object recognition memory, fear and emotional memory, and spatial memory as well, which is a really, really big thing. And we came up with some really, really cool results. We found that the CBD had kind of no effect on novel object memory retention, but that's not a bad thing because we didn't have a negative effect, which is really, really good to see. But we also found that CBD actually improved spatial memory, and it also ended up improving a fear-based memory that we find in the amygdala. And then overall, after we kind of went through the histology and the brain slicing, we also found that CBD actually ended up increasing the amount of cells in the hippocampus. So overall, we found that CBD actually plays a really, really strong role in improving a variety of issues that occur when individuals age. So yeah, hopefully we can do more of this research in humans. Thank you.
Thank you, Maliha. Thanks for your work. Can I talk to the next trainee, David? Hi. Tell us about you, where you're from, and your project.
All right. So my name is David Robertson, and I am with the University of Lethbridge with the McDonald Lab. And my research is looking into the effects of circadian rhythm disruption on the hippocampus. When I say circadian disruption, I'm talking about shifts in the time of day when we go to sleep and when we're exposed to bright lights, and the way that shift from what our circadian rhythm expects can have impacts on our learning and memory. Now, previous studies have found that these circadian disruptions can negatively impact certain learning and memory functions related to the hippocampus. But those have been behavioral studies, and this one is looking specifically at and directly at the place cells within the hippocampus. So we've been taking actual videos of the firing patterns of these neurons and have been analyzing them to isolate which ones are the place cells, and then taking a look to see whether the patterns of that firing has been altered or disrupted or shaken by the circadian rhythm disruptions. the whole focus of this is to take a look at the memory and how the memories are either strengthened or weakened by disruption to the circadian rhythm. And so we're still in the preliminary stages, but excited to see the results.
Thank you, David. Thank you, David. Can I talk to the next trainee? Matt. Welcome, Matt.
Hi, I'm Matt Nielsen. This was work that I did with Dr. McDonald in my undergraduate degree for my honors thesis at the University of Lethbridge. It was kind of a different version, measuring in circadian disruption, but instead of doing it with light patterns, we decided to do it with keeping the same day and night schedule, but just offering a sugar meal before bed. So we would take all of our adult age rats and we would put them in their separate housing, and then some we would feed a regular diet, and the others we would also feed a regular diet, but just before their bedtime, which would be the daytime for us, we would feed them a high sugar meal, and then we measured their activity, and then after a long period of time we decided to measure their spatial memory using a spatial memory task. So we decided to look at the difference between the males and the females. And in the males, we found a deficit in their long-term memory. In their short-term memory, they managed to achieve the spatial memory task, but after an extended period of time, we began to see deficits. And then the other interesting part is when we measured a female group, we saw that there was no deficit, which pointed to two things we've been working towards in the body of information in hippocampal memory during circadian disruption, which is that there is a sex difference, as was mentioned by Dr. McDonald, that females have this sort of innate resilience built in, and where that comes from is not quite known at this point. But then exemplified in the males is that all of these groups were kept on a same day-night cycle. So that's the equivalent of getting a good night's sleep. You're getting your average 8 hours. You're getting, the exact time that you need. But the only difference is just taking that sugar meal before bedtime can have that memory deficit. So for the equivalent of the student who is getting a good night's sleep, even something as simple as having a lot of sugar before bed can affect how much you remember for that test in the oncoming weeks. So yeah, there's still more data to process, but the preliminary results look really good.
And what school you're from, Matt?
University of Lethbridge.
Thank you, Matt. So no sugary snack is better for memories? Is that the...
Maybe once. As with circadian rhythms, you train it with, continual use. You train your sleep patterns. So the odd bad night here and there won't be too bad. So if you're really feeling it, maybe once in a while, but regular.
I like chocolate before bed sometimes, so that's why I'm asking.
Yeah, no, once in a while, maybe,
Thank you, Matt.
Maybe not.
Thank you. Hello.
Hi.
Tell us your name, where you're from, and about your project, please.
My name is Jinjing Yao. I'm from the University of Calgary, and I'm working in Dr. Win Chen's lab. So what we are working on is we're trying to find out what the real cause of the Alzheimer's disease. As Dr. McDonald mentioned, there are about 95% of the AD patients are sporadic. So we don't know the real cause of the AD in these patients. So previously of our studies shown that in the AD brains, the neuron actually become more active. So another disease called the TIA, aka called small mini stroke, which is a small stroke happen in your brain. You may not even sense it, maybe a couple days of headache. But we found that this kind of stroke can also increase your brain activity. And from the slice work and also the animal work, we found that simply trigger the TIA can trigger the similar thing happen in the AD mouse brain. And we found that controlling the calcium release in the neurons actually help us to overcome this kind of damage caused by TIA or Alzheimer's disease. And we have a very powerful drug can treat these diseases.
That is so cool. Thank you very much. And next?
I got this little computer action. It's not quite a poster, but maybe you can see that at home. My name is William Betzner. I'm a researcher, Master of Neuroscience student here at the University of Calgary, and I'll give you a brief walkthrough of this kind of interesting mixed methods study that we're conducting over here again at the U of C that's looking to explore the experiences of people living with dementia who have a stroke. So we know there's lots of Canadians that are affected by dementia, and similarly many of these elderly individuals might experience a stroke, and it's about 10% of stroke patients that come in with dementia, yet we often exclude them from both routine care and clinical trials, meaning we have this big data gap. We don't know how these people really fare in acute stroke care, and we are not super sure on how to treat them. So to address this, we're doing this mixed method study that incorporates 2 sets of interviews, one with patients and their primary caregiver, a second with physicians, and both those are feeding them into a survey-based study that kind of helps paint a really holistic picture of acute stroke care for people living with dementia. So that's kind of the aim of this project. Both the interview studies are underway. We found some really interesting findings like issues with first responders, the need for clinical trials, the importance of patient prognosis, desire for independence, lots of really interesting themes coming from those interviews.
Thank you.
No problem.
Very neat. Can I get you to introduce yourself and tell us what school you're from? Well, I can see.
Yeah, it's a huge poster. So, my name is Fernando Vega. I'm from the University of Calgary. I'm a master's student. My project is amyloid beta PET synthesis from a structural MRI, a potential alternative method for Alzheimer's disease screening. So, just as we saw, Alzheimer's disease can be detected with amyloid beta and brain atrophy, which can be targeted with positron emission tomography and magnetic resonance imaging. However, performing PET scans is quite difficult. The problem with PET scans is that, despite that they provide molecular information, they are real expensive, 10 times more expensive than MRI. And the problem is that it involves injection, and I don't know about you, but I don't like being injected. And it also induces radiation to the patient because this radiotracer, it's a radio-labeled compound. On the other hand, MRI is a cheaper technique, where it's less expensive, it's a non-invasive, it's free from ionizing radiation. So what about if we use artificial intelligence to make PET images from MRI? Therefore, we reduce the cost, we reduce the radiation exposure, and we make this technique more available in places and clinics where a cyclotron is not possible because a cyclotron is really expensive. So that's my research. As you can see in this huge image, is that I have the MRI, the PET, that was the original PET, and this is the one that my artificial intelligence created. And this one is just a difference between these two. That's it.
So this was created by AI, and normally we would have to pay a lot of money to get this scan done, right?
Yeah, exactly.
A PET machine or PET image versus using the MRI, right?
Yeah, so, yeah, you're correct. But the thing is just like we use MRI alone, and then we infer the PET.
Oh, okay. Thank you.
Yep.
Hi, introduce yourself. Welcome.
Hi, I'm Jerry. I'm a student at the University of Calgary, and my study is on a systematic review assessing the effectiveness of art, music, and recreational therapy on systems and quality of life of persons with early onset dementia. So, unlike normal dementia, which occurs in patients in their 40s and 50s, early onset dementia manifests before the age of 65. So we're trying to assess the effectiveness of these non-pharmacological interventions on the quality of life of these patients. So we conducted a systematic review using the inclusion criteria and the exclusion criteria in Table 1, and we involved individuals with early onset dementia and clinical trials, and it's published in English. As for exclusion criteria, we excluded the studies that involve the wrong study design, or systematic reviews or interventions that consisted of recreational therapy outside of community-based settings. So based on our findings, we obtained 0 studies that assess the effectiveness of these non-pharmacological interventions, because most of the studies did not have results that were separated by age. And even then, one study that included 2 stratification groups, which are mainly 45 to 55 years old, and 56 to 65, they only included caregivers of individuals with Alzheimer's disease. And it doesn't exclusively say that these patients are of early onset dementia. So based on our findings, we can say that the studies based on the effectiveness of art therapy, music and therapy, and recreational therapy is limited, so we have to conduct more research so that we can focus on supporting the maintenance of functional abilities in individuals with early onset dementia. Yeah. Thank you.
Thank you very much. And I think that is everyone, all of our trainees. Thank you so much. Thank you everyone who attended our event this evening. And the Alzheimer's Society of Alberta and the Northwest Territories is here for your education. If you need support, if you have questions, we're at 1-866-950-5465. And online, you can reach us through our website, alzheimer.ab.ca. Thanks so much and good night.